Differences in electrochemical gradients determine the resting membrane potential. This section is a lot of details on different ions, but it's really important to understand this section in order to understand how an action potential travels and is propagated. So there are a variety of contributors to the resting membrane potential. Think of your cells as being in a seawater bath. That means outside the cell, there will be a lot of sodium chloride or sodium and chloride ions. So the extracellular fluid is high in salt, sodium chloride. Inside the cell, there is a high concentration of potassium ions. The cell actively pulls potassium ions in them, and there are many negatively charged proteins. Now, the proteins cannot escape the cell. They're stuck in there. The sodium and potassium ions are the main factors influencing that membrane potential. That is what is giving it the negative charge inside and the positive charge outside. These ions cannot move freely across the cell membrane. They have to move by membrane channels or active transport, which would require ATP. The resting membrane potential is a form of potential energy. It's measured in millivolts. A millivolt is 1,000th of a volt, and the body cells have a range of millivolts. The resting membrane potential of a neuron is negative 70 millivolts, and that negative value is just because it is more negative inside than outside the cell. The remaining resting membrane potential involves passive forces and active forces. The passive forces, passive meaning no ATP is required, include diffusion of sodium and potassium through channels called leak channels. Also, another feature are the negative proteins inside the cell, and those do not leave the cell. The active process is the sodium-potassium exchange pump. You may recall this from previous chapters, but we'll be getting it again in this chapter. Leak channels are always open. The ions that pass through are determined by that particular leak channel. So we're interested in potassium leak channels and sodium leak channels. Potassium ions leave the cell through potassium leak channels, and sodium ions enter through sodium leak channels. Keep in mind the cell is in seawater, so there's always more salt outside, so the sodium tends to leak to the inside. The active mechanism is the sodium-potassium exchange pump. Well, because sodium is always leaking in, the cell wants to get rid of it. On the other hand, the cell wants more potassium, and yet there's not that much outside the cell. There's much more potassium inside the cell than outside the cell. So the sodium-potassium pump gets rid of 3 sodium ions for every 2 potassium ions it brings in. And this maintains the stable resting potential. So you can see a variety of things going on here. We have our negative proteins inside the cell. There are positive charges inside the cell. There are positive and negative charges outside the cell. But relative to each other, inside the cell is more negative at negative 70 millivolts than outside the cell. There are leak channels. Potassium is always leaking out. Sodium is always leaking in. And to help maintain this balance, we have the sodium-potassium pump that shoots out 3 sodium for every 2 potassium it brings in.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
So there are two potentials. There's the chemical gradient. There are two gradients, the chemical gradient and the electrical gradient. The chemical gradient is the concentration gradient for the ion across the plasma membrane. For example, there's more potassium inside the cell than outside the cell. There's a gradient there. The electrical gradient is created by attraction of opposite charges or repulsion between like charges. Just as with a magnet, north and south attract each other, but north and north poles repel each other. The electrochemical gradient is the form of potential energy determined by a combination of the chemical and electrical gradients. Equilibrium potential is the membrane potential at which the ion's electrical and chemical gradients are in balance. There will be no net movement of ions across the membrane. The equilibrium potential for potassium is negative 90 millivolts, and for sodium is positive 66 millivolts. For the potassium ion, at the resting membrane potential, the smaller electrical gradient brings potassium into the cell, but the larger chemical gradient takes potassium out of the cell, so the net gradient is out of the cell. Sodium is different. Sodium, both the electrical gradient and chemical gradient are directed into the cell, so therefore sodium has a very strong net gradient into the cell. Which term is used to describe a transmembrane potential in which the electrical and chemical gradients are equal and opposite with no net ion movement? That would be equilibrium potential.